Let's look at carbohydrate digestion. Carb digestion begins in the mouth with secretion of salivary alpha amylase. Salivary alpha amylase, like all alpha amylases, will randomly digest only alpha-1,4 bonds. And the products of that little tiny bit of digestion that we have in the mouth are kind of chunks of carbohydrates that are called dextrins. There's no digestion in the stomach, but what happens to the carbohydrates in the stomach is that our viscous fibers, which is a subcategory of soluble fibers, can swell up. So our viscous fibers can swell up and they can bind. They can bind sugars, triglycerides, fatty acids, cholesterol, and some of them can help increase satiety or that feeling of fullness. If we move on to the small intestine, we have one pancreatic carb digesting enzyme that's called pancreatic alpha amylase. And pancreatic alpha amylase is just like the salivary alpha amylase. It just has more time to do digestion, but it's going to randomly digest only alpha-1,4 bonds. So since we have more time, our products are going to be smaller chunks. It's going to be glucose, maltose, and isomaltose. And then once we get it digested into those smaller chunks, then the brush border enzymes, which are known as disaccharidases, are going to take over. Some of the disaccharidases include maltase, lactase, isomaltase, and sucrase. So your maltase is going to break down maltose, which we said on the previous concept map, maltose is glucose, alpha-1,4 glucose. So we're going to end up with 2 glucoses. Lactase is going to break down lactose, which is a mixture of galactose and glucose. So you get some galactose and some glucose. Isomaltase will break down isomaltose. If you recall that isomaltose is an isomer of maltose, which is glucose alpha-1,6 glucose. We end up with all glucoses from there. And sucrase will break down sucrose. Sucrose is a mixture of glucose and fructose. So you can see we have a lot of glucose, a little bit of galactose, a little bit of fructose coming in there. So once we have it broken down into those single sugars, our monosaccharides are what we have to digest it into, because that's the largest sugar that we can absorb. We're going to absorb our carbohydrates in a couple different fashions. We can absorb our glucose and galactose are going to be absorbed by active transport. Glucose and galactose are absorbed by active transport using SGLT1, that's sodium glucose transporter 1, which requires sodium, potassium, and ATP to run that sodium, potassium, ATPase pump. We can also absorb some by facilitated diffusion. The carriers that we have for facilitated diffusion are GLUT5 and GLUT2. GLUT5 absorbs only fructose, and GLUT2 can absorb all of them, but we're looking at GLUT2 only being inserted into the membrane after carbohydrate consumption, but before insulin release. Insulin will pull GLUT2 out of that brush border membrane. GLUT2 will always be in the basolateral membrane because it's important for transport out of the enterocyte. So our carbs, all of them are going to be transported into the portal vein because they are water soluble via GLUT2. So again, that's all of our sugars are able to bind onto GLUT2 out through that basolateral membrane through facilitated diffusion and go to the portal veins to the liver. Now the liver is going to control, get a bunch of the glucose coming in. Our normal fasting blood glucose concentration is 70 to 99. That's milligrams per deciliter. And our blood sugar is going to be controlled via 2 peptide hormones that are both made in the pancreas. The first peptide hormone is called insulin, and that's released from the beta cells of the pancreas. And that's secreted in response to elevated blood sugar, so high blood glucose. It can also be elevated in response to arginine. and CCK helps stimulate release of it. Insulin's your builder. It's going to help build your glycogen. It'll build triglycerides. If you have excess carbohydrates, we can turn them into fat in the liver. And we can also help build muscle. So we're looking at storing carbs, lipids, and proteins. The opposite end of the spectrum is the other peptide hormones released is called glucagon. Glucagon is released from the alpha cells of the pancreas, and both your alpha and beta cells of the pancreas are releasing these hormones, these peptide hormones, which means these are endocrine cells. Glucagon is secreted in response to low blood glucose, so decrease in blood glucose, as well as during our fight or flight mechanism, so epinephrine can help stimulate release of glucagon. Glucagon's your breaker. It's going to break liver glycogen in particular, help increase blood glucose. It'll break down triglycerides if we need more energy, and it can break down muscle for gluconeogenesis if we absolutely need it to. We'll release those glucogenic amino acids to be used for glucose in gluconeogenesis. So let's say we have an imbalance in our blood sugar. Our body can't figure out how to regulate our insulin and glucagon. If we have uncontrolled diabetes, we have several different things that's going to happen. For one, short-term blood glucose regulation, you can check glucose concentrations throughout the day, but your long-term marker of blood glucose control is called HbA1c, or hemoglobin A1c. This is a type of compound where you have it's a protein and a sugar attached to one another. It's called an AGE, or AGEs. It's called advanced glycation end products. These things are irreversible, so they're stuck together, and we have to either metabolize the whole thing or they will accumulate. If we accumulate them, then we get complications. So these complications, if we get stuck in large blood vessels, are called macrovascular complications, which would be cardiovascular disease. It kills 75% of all diabetics with uncontrolled blood glucose. It's cardiovascular disease. If the HbA1c gets stuck in these small blood vessels, it's called microvascular complications, and that can lead to nephropathy, kidney damage, neuropathy, nerve damage, and eye damage, retinopathy. So it's very pertinent that we regulate our blood sugar, get it under control, keep it in that 70 to 99 range during fasting times. So otherwise, for every little 1% increase that we have in HbA1c, we have a 10% increase in our microvascular complications. To help have a minimal impact on blood glucose, we can consume more fiber. That can help bind it, especially the viscous fibers, and help level it out a little bit more. There's that thing called the glycemic index. There's artificial sweeteners, and we have some sugar replacers. With sugar replacers, that would be things like tagatose, which is naturally found in dairy, and sugar alcohols. Sugar alcohols are sugars that are reduced, so they're made by reduction. And with tagatose and sugar alcohols, they do provide a little bit of energy, which is different than the artificial sweeteners. So we get, again, about 1.5 to 2 calories per gram because we can ferment tagatose and the sugar alcohols. Since we can ferment them, side effects include explosive diarrhea. So gastrointestinal disturbances. Your artificial sweeteners provide 0 calories per gram. A little bit different. And again, that if they're fermentable fibers, 1.5 to 2 calories per gram. Okay, that's the end of our carb digestion. Thank you very much.